right, welcome everybody. So I'm going to talk about some work that's been over 5 years in the making because I live or work in a big, huge organization now. So we'll see how this takes place. So once again, here's my background. One of the things that wasn't on the flyer was that I was a community college student at Fullerton College in Fullerton, California, which is just north of Anaheim, California, which is where, what is? Disneyland is, okay? So I wanted to let you know that. So that's Orange County, of course, in California. I went to Cal Poly. I switched after 2 terms from being a biology major to a biochemistry major. So you can change your mind and it will work out in the end, okay? So make sure you have a plan. When I was doing my PhD, it's in pharmaceutical sciences, so with a minor in chemistry. So basically I had some pharmacy classes I had to take, drug design, drug synthesis, and advanced organic chemistry. And all those things come together in what I'm going to be talking about today. And then I did spend some time in Texas as well on a postdoc. Okay, so my official title at work is a medical affairs scientist, which means that I do some science and I also deal with the FDA. So what you're going to be seeing here is that I helped originate this idea, and then I also helped shepherd it all the way through to getting an IND, which is an investigational new drug application, so that we can put it into patients. And if you do government work, you always have these acronyms here. So ADRD, LBR, and FNLCR all correspond to these things here, my directorate. I'm embedded in the headquarters of the National Cancer Institute, and so I provide support for CIP, DCTD, NCI, which is part of NIH, and I actually work at the NCI headquarters, which is on the Shady Grove campus at Johns Hopkins. So it's just up the street from Shady Grove Hospital. If you go around this way on Medical Center Drive, you'll find us, okay? So that's what we do. All right. So, of course, Leidos Biomedical operates the Frederick National Laboratory for Cancer Research. It's a federally funded research and development center. It's the only national lab in the Department of Health and Human Services. It's located primarily on Fort Detrick, so if I need to go to do HR things or if I visit my radiopharmacy, that's up in Fort Detrick. And I work for a prime contractor, and that makes me a pseudo-fed. And if you don't get that as a joke, well, I'll tell you it's a joke, all right? And all my pharmacy jokes are the lamest. Okay, so clinical question. One of the doctors, actually the associate director, came and said, okay, we know that HER-scan works in the laboratory, in the clinic, and I want to know if it can be extended to EGFR monoclonal antibodies. This comes from, trastuzumab comes from a related family. It's HER2 and EGFR is actually HER1. So yeah, we said, yeah, probably can work that out. So here's the background. Cetuximab and panitumumab are monoclonal antibodies, and they target EGFR or HER1. And the trastuzumab goes for the HER2. This is your molecular biology for the day. These bind to a receptor and inhibits phosphorylation and activation of a kinase, and it results in inhibition of cell growth and downregulation of growth in the overall tumor. But what we want to use these for is, since these are therapeutics, we want to transform those into imaging agents by adding something to them. Hopefully not too much to make them inactive, and seeing if naive patients will have these receptors in their body, and then we can know whether to continue treating them with the therapeutic or not. So EGFR is in lots of cancers or lots of cells, so lots of cancers will be expressing this. It can be especially found in colorectal cancer, which is one of the ones that we don't really have good cures for at the current time. So the molecular trafficking and fate of trastuzumab, cetuximab, and panitumumab seems to be very similar. So the answer to the first question, by extending the radioligand approach to the EGFR, was I gave the boss a yes. So I started this all a little bit after I got here in 2007. So in 2008, cetuximab had been on the scene for quite a while, but panitumumab was just getting approval from the FDA. So the easy question first, which of these monoclonal antibodies will make a better imaging agent? And of course, you have to make both of them and try it out, but one should be better than the other. And the tougher question that he asked me to look at later in 2008 was, since the molecular imaging program at the clinical center down in Bethesda only has PET capability, positron emission tomography capability, can I convert it from SPECT in the current agent to PET in the future agent? So that means to do actual research. So what is SPECT? SPECT is detected using gamma rays. You have a gamma camera, you put the agent that's radioactive into the patient, you put the camera near them, and the camera detects whether they have localized disease or not. And you have to have a gamma-emitting radioisotope or radionuclide, and you put it in by injection. The ones that we use, we have to attach to the protein a radioligand and then attach the reactive radioactive metal on there. And then we hope that that's targeted to the patient so we can see where their disease is. Now, using SPECT is not as accurate as using PET. So on PET, usually you get a PET CT, and those two things together will give you a lot of information. So what we do is we have a PET agent, and we put that into body, and then we have a PET camera. And in SPECT, there's only a gamma ray that's given off, and it's detected. But when you have PET, you have a pair of 511 photons that are given off in opposite directions. And that is actually what the PET detector is detecting, is the coincidence of those two particular things. And what we would like to use for that is that everybody who gets a PET CT scan, usually it's done with FDG, which is fluorodeoxyglucose. And it lets you know where the cancer metastases are, because by the Warburg effect, it goes where there is glucose being utilized. However, for our purposes, we need something after they get their FDG scan to say, well, where is their disease, and do they have metastases elsewhere in the body that are specific for the targeted drugs that we have to use against it. So we have our therapeutic antibodies, and we chelate them, or we bind them, conjugate them to a chelating moiety to bind the metal. And we need to demonstrate that we have in vivo distribution, that we can go after the potentially sensitive tumors, that we have downregulation after treatment. We have to provide dosimetry for imaging to show that the FDA that we say it's going where we want it to go and not anywhere derogatory to the patient, to give us a safety signal. And we might be able to select patients using imaging. All right, so the real trick is to use the items from nature's toolbox, given the following parameters for making imaging monoclonal antibodies via chelation with radioactive metal ions. So full-size monoclonal antibodies have a long half-life. They take a long time to clear from the bloodstream. That relates to that. Full size are best imaged between 2 and 4 days after administering the drug. So if you give it IV, you need to wait between 2 and 4 days for the blood to clear, and so you're going to be able to see what you need to see. Full-size monoclonal antibodies are temperature sensitive and condition sensitive. Radiometals have various PET half-lives, and radiometals require proper size for good chelate fit, and excellent matches can be difficult to make. So this was a known compound that was invented by somebody else down at the clinical center, and it was actually it's manufactured by a company under license. And this is what they used to attach to the trastuzumab in the first experiment. And you get a nucleophilic attack right here on this carbon, and you make a new amino carbon bond, nitrogen carbon bond that you'll see later. And then you can stick your radiometal is attracted to this particular portion of the molecule. Notice we have 1, 2, 3 nitrogens, and we have 1, 2, 3, 4, 5 carboxylic acid groups, so that's why we have an N3O5 motif. And if you put this in your Spartan, you can see how it wraps around. Here's our indium in this particular case, because that's the metal that was used. And you can actually see through this is a carboxylic acid group here, here, and here. You can't see the nitrogens because they're on the back part, so you'd have to look through the whole thing. So it doesn't completely cover up the radiometal, but it binds pretty well. You get a stability constant of 29 and for the indium Chicxay, and for the zirconium Chicxay, it's about 35.8. So this was the existing one, and this was the one we wanted to make, so that looks pretty promising. So this is how you have to make this stuff. You have to get your panitumumab in a sealed vial from the pharmacy, and then you have to prepare all of these buffers that you're going to use and demetallize them so there's no trace metals that are going to interact with your chelator that you're going to use later. And you bind your panitumumab to your chelator. You end up with your panitumumab Chicxay right here in this particular case. And you end up with attaching the indium after you put the radioactive label in there. So long story short is you can make the stuff that you need, and you can see if you can image it. So here's a schematic of the same thing, and what we see is these particular cell lines. This is a low expressor, this is a medium expressor, and this is a high expressor of the HER1 or the EGFR, and that kind of correlates to what was seen when we did the panitumumab with the indium-111. So the uptake did correlate with the radial labeling with EGFR protein expression, so we thought we were on a pretty good track. So we can actually make the SPECT agent for this, and we could use it for imaging. And the trick here is for you to tell me which way the mice are rotating, clockwise or counterclockwise. What we do is there's another fellow out in Frederick who puts some tumors under the skin, and that's what you see for the hot spot here and there and here. And as you can see, there's uptake in some of the other organs, but the tumors themselves are actually pretty well lit up. And this is what you get on a SPECT image. And the answer to the question is whether it's clockwise or counterclockwise, it depends on which eye is dominant at which time. I can actually look at them and flop them back and forth. All right. So that's what an imaged mouse looks like. All right. So the first question we could answer was yes, we could do that. And so we needed to work on translating it to PET from SPECT. So we talk about the radioligand and a radiopharmaceutical that we're going to produce. In this case, we need that radiopharmaceutical to go from a SPECT pharmaceutical to a PET pharmaceutical so we could use the facilities that were down at the clinical center in Bethesda. So I had to go online and do a lot of research and find out what was commercially available or academically available. And you can get several coppers, you could get titanium, you can't anymore, technetium, gallium, and yttrium from WashU in St. Louis. And they were available, a lot of these are in-house only, so some of them, the Y86, they would ship to us. The titanium turned out that it was toxic, so we didn't want to use that. And the Y86 has problems with something called Compton emissions that interfere with the PET detection some. So I looked there and said, okay, well, if we have to do a backup, it's going to be this one. I went to Oak Ridge National Lab and found out you could get radioactive arsenic, vanadium, a different yttrium, and some iron. They say that it's available monthly, but you have to special order it, and they will tell you, well, we'll get to it, we'll get to it, we'll get to it. Oh, okay, well, whenever. Other agents that have been used in other research auspices are these ones here. We got a scandium, a cobalt, a germanium, and another arsenic, but I didn't have a readily available source in the United States for those. These have been done in Europe. So that left me with zirconium 89, gallium 68, and copper 64, all of which are available. And I put the indium 111, which is the spec reference, in here so that we could compare. So it turns out these three are all PET, so that's good. This is the average positron value. So the gallium one is the hottest one, has the most energy, and the copper is next. And our zirconium's not too bad. It has a gamma at 909, but these have even higher gamma, so this one's definitely the most attractive. And then the final decider was the half-life. So if a monoclonal antibody is going to be around for 2 to 4 days, you've got to have a radioisotope that's going to be around, that's going to have a half-life of 2 to 4 days, and certainly a half day is not going to cut it. The gallium definitely wasn't going to cut it. This is like 1 hour or so, and looks like we're going to be stuck with our zirconium. So they had started marketing zirconium 89 in Europe at the time, and we actually ordered it from a company called IBA. And the second time we ordered it, it got lost at Dulles. It never made it out of Dulles, and so we said, that's not going to be reliable. So we said, okay, well, let's talk about the actual chemistry or the size to help us choose also. So our indium for the DTPA is 0.92, and so the zirconium is about the same, and we know from the stability constant that it's going to be pretty close to that. This one's a little small, that one would be okay. Different charges, configurations, and coordination numbers for each of those. So turns out shortly after we started this that they started making zirconium oxalate, zirconium 89 oxalate, in the cyclotrons at the clinical center, so we could get some, buy some, send it up to Frederick, process it into our particular stream, and send it back down. So we were happy then because we didn't have to rely on IBA's reliability to get it here, which of course we said was suspect. So this is what the zirconium oxalate looks like. There is one zirconium here in the green, and there are 4 oxalates, each one contributing 1 oxygen here. And so I got this off the crystal structure off the internet from a reputable place. You can see that the stability constant's about 22, which is less than the stability constant for the one that we are going to be transferring it to. So that's the space-filling view, and here's the density potential map. And so this is how much of the zirconium is actually available to, surface is available to be bonded to crack this open. And once again, here's our complex scheme to get it all on. And even though it's produced and delivered at the clinical center, the zirconium oxalate does not transfer zirconium to the CHICSA DTPA, so our easy experiment went down the tubes. Why? Well, the N3O5 motif is not able to steal the zirconium from the O4 motif of the oxalate. It's not strong enough to steal it away. So can't crack the acorn, or whatever you want to call it, or the walnut, and get in there and take it out. However, all is not lost. The oxalate can be converted to the zinc zirconium chloride, and that should make it more accessible than the oxalate and have a lower stability constant, which in fact it does. And this blue patch is slightly higher or slightly larger than the blue patch on the oxalate, but when my guys in Frederick tried it out, it doesn't transfer either. So we're thinking, okay, well, zirconium is a little bit harder in terms of a radiometal than the indium, which is kind of softer, and you just can't break it out. So neither of these approaches to make the easy stuff is going to work for us. So now we have to have something that the zirconium is going to go to, so we had to switch chelators. All right, so if we switch the chelator to desferoxamine B, okay, or DFOB, or desferal, it's all the same name for the same thing. It's interesting that desferal is a drug on its own. It's given to patients to remove excess iron, and it's made by bacteria as a siderophore. A siderophore is a product that is secreted by the bacteria to go and get resources from outside, and then it comes back in. So it secretes the DFO, it goes and fetches the iron, which the iron comes back in, and so the bacteria can use it to eliminate its neighbors. So here's what DFOB looks like, and here's what DFOE looks like. Notice this is fully cyclized, this is open chain here. But it's an O6 motif. So the oxygens involved here are this one, and this one here, and this group right here. So if you're looking at the iron complex of this, that's what this looks like. So like I said, the bacteria would secrete this without the iron in it, go get the iron, bring it back in, advantage to the bacteria that can handle that. So the people that made the previous chelator also were progressing at about the same time, so they have provided this activated intermediate, which we can use, that has the same functionality or reactive functionality here. Even though the chelator is different, the business end of making the molecule is over here initially, so that we can bind it to our proteins. So we can order that from macrocyclics, and we're really happy about that. They call it desferoxamine para-isothiocyanate. Okay, so we could use that. We get our nucleophilic attack by a lysine here and make a new bond. So what does the zirconium look like? Zirconium DFO look like space filling. You can see this is a zirconium here. This is our NO. This is the one that's deprotonated, so we can see that it doesn't cover up the zirconium completely. But the indium is a lot lower, and the zirconium is pretty good in the DFO in terms of the binding constant, so we're happy there. And if you look at the density potential map, this is exposed to a little bit of water. So you might get a water molecule that binds in there. Once again, we go through that same procedure, but this time we're going to use our DFOB and attach it to our panitumumab. And of course, now we have something that's going to bind to our zirconium oxalate, and we know for a fact that this will work. The desferoxamine is much stronger. Since it's an O6, it can bust out the oxalate and take it away from the oxalate. And so here's the schematic where we have a free amine. We get a nucleophilic attack here, form a new bond. This is a thiourea bond, which is not one of your common organic functional groups. And then the rest of the molecule is, of course, over here, and we're going to react our zirconium, and it's going to bind to that. The importance is that thiourea linkage is stable for more than 2 to 4 days in a human. So that's what we're hoping. This shows the ratio of how many DFOs they put with the monoclonal antibody and how many they actually got on there. So it's bilinear here. And this is what the final bioconjugation looks like, where we have our radioactive metal bound in here. All right. So we characterize that by doing a couple procedures up in Frederick. We have our panitumumab. We put it in the HPLC. We know where the peak for the panitumumab goes. When we have our zirconium panitumumab before purification, we have 4 peaks corresponding to the oxalate, the unreacted chelate, some of the panitumumab, and some of the zirconium 89 panitumumab. And after we purify it in the HPLC, we get one peak, so we know we're making a good product. We bottle that up and send it down to the clinical center. We can also use something called ITLC, which of course is just thin layer chromatography. So don't think the skills that you learn in organic go by the wayside. We have to use a phosphor imager to see the radioactivity on there, but it is just TLC. So they use both of those methods up there. All right, so now we have some new cell lines that we want to try it against. BT474 is the lower expressor. That'll be the negative control. We have a moderate expression with the MB231s, and we have a high expression with MB468s. So we needed to do some more in vitro studies, and I'm not going to spend that much time on these. And we end up getting a decent biodistribution. Here's our panitumumab with the indium label, and the zirconium label is a little bit higher, but it's essentially the same. So we said, okay, we're good. All right, I'm not going to talk about that one. This is just the biodistribution is the same. So we know they're both going to work, and this is what the dosimetry results look like. This is the type of data that you have to furnish to the FDA in order to say it's okay to put this in humans. But you have to sacrifice a couple animals in order to do this. So they actually inject it and then sacrifice the animals and then analyze each one of these tissues separately. So we have that information for the indium and the zirconium. And it was found that both tracers are in the axillary lymph nodes at later time points, and we don't think we're going to see that in humans. All right, that one I'm not going to talk about. So this is the result that we want to discuss. Despite the higher per unit radiation dose, given the higher sensitivity and superior potential quantitation of PET compared with SPECT, we chose to pursue our zirconium labeled radiotracer, and the subsequent imaging studies were performed only using the PET agent. So you can rest your eyes on this one. This one doesn't rotate. Here's our BT474 tumor right there, and as you can see, it's not very yellow compared to that one. So the low expressor doesn't show up very well, so that seems fine. Our mid expressor, yeah, it's a little bit brighter here, so that correlates with that. And our high expressor, we get a pretty good signal from. So it correlates in vivo with what they found in vitro, and so we were able to use this information to go ahead and go on to the next particular type of thing. This is another slide that they want to see, correlation for that. So, let's see. That's just information from... All right, let's concentrate on this one. EGFR protein expression levels were obtained from the excised tumors, and the resulting strong correlation, 0.857 of the zirconium panitumumab uptake with the EGFR actin ratio. That's the result that we want. We would want this over 0.90, but we'll take what we can get. And it says that the different cell lines, and because we have correlated with them in such a way that we can go ahead and use this as a promising radiotracer. So the high in vivo stability and long half-life of the desferoxamine with the zirconium allows accurate measurement of the panitumumab pharmacokinetics, and that can't be achieved by other well-studied PET isotope agents, so we ruled those out. It's prepared with high radiochemical purity and specific activity, and we don't lose any of the radioisotope in serum. That's especially important because you don't want radioactive metals wandering around in your patient. You want them to be maintained and excreted appropriately. Okay. All right, so we went ahead and did it. So let's look at the answers here. So at the time this all started, cetuximab had been available for several years. Panitumumab was coming on. The panitumumab imaging was much better than the cetuximab imaging, which has also been borne out in the clinical history of these agents over the last few years as well. So, could I get the molecular imaging program what they needed and convert that from SPECT to PET? And the answer to that was yes. And so then I had more work to do. So let's go ahead and see what we had to do. So in order to progress to the clinical trial, I had to get permission from the NCI Office of the Director in Frederick. I had to get permission from my biggest boss. We had to go to the Nuclear Regulatory Commission and make sure it was okay with them. We had to go to the Maryland Board of Pharmacy since we were preparing a drug. We had to go to the radiation safety offices both in Bethesda and Frederick because they have to correlate or they have to coordinate activities. We have to arrange for proper transport. You can do it by FedEx, but it's a special FedEx guy that has to do it because there are DOT, Department of Transportation regulations. We have since hired another special contractor, which is cheaper than FedEx, to run it from Frederick down to the clinical center in Bethesda. And we had to get permission to put it in patients by filing an IND for the drug. So, IND 101. If you want to put it in somebody, you need an IND to make sure that it has the safety. You've documented all the safety requirements. So not only did I get to originate the idea, but I also get to help shepherd it through this particular process as a medical affairs scientist. So when a product is identified as a viable candidate, the sponsor has to focus on collecting the data and information necessary to establish that the product will not expose humans to unreasonable risks when used in limited early-stage clinical trials. All right. So, the IND application must contain animal pharmacology and toxicology studies. That proves that it's safe in animals, and we project that into humans. We have to supply the manufacturing information. So every single procedure that goes into the making of this, each SOP, standard operating procedure, has to be listed and then compiled in something called the CMC, which is the chemistry and manufacturing controls, and submitted for inspection to the FDA. This information is assessed to ensure that the company can adequately produce and supply consistent batches of the drug. So the clinical protocols and investigator information was done by the people at the clinical center, and they have to submit that through their own things, something called an IRB, which is an institutional review board, and they have to get permission for that. So the IND was filed on 9/19/12, a little over 2 years ago, or coming up on 2-year anniversary. Patient accrual stands at 3 human subjects, and we're going for 20. And they have since developed another study that can use this agent. And we're going to hopefully have 45 patients in this particular study. So, acknowledgments. The big boss on this Fed side is Dr. Dorshow. He's down. He's one of Dr. Varmus's, who's the head of the NCI. He's one of the deputies for Dr. Varmus now. The one who gave me the challenge was Dr. Tatum and Dr. Jacobs. And a small animal imaging program was Dr. Joe Kalin and Lisa Riffle. So those nice images of the mice that you saw, they were produced by these two. My radiopharmaceutical group, Siba Bhattacharya and Ling Wei, they do most of the biological stuff. Out at Frederick also, Dr. Hollingshead and Dr. Carr did the tumor and the westerns for us. And our colleagues down at the clinical center, Dr. Choiki and Dr. Kurdzile. The radiopharmacy, the people that actually make the doses are George Afari and Xiao Xi. And then in our area, myself and Ms. Ugas are the ones, she's the one that compiles the information and publishes it for submission to the FDA. So here's my disclaimer, or I have to have this to say that this was funded by NCI according to this grant number. And doesn't imply endorsement by the US government. I speak for myself. And if you want the reference, it is currently found in Nuclear Medicine and Biology, volume 40, pages 451 through 57, and it was published in 2013. And that is the end of the show. Thank you very much.